Hello everyone, hello mister. I hope you all are having a wonderful time. Today, we're going to present the laboratory number 5 of food microbiology. We're going to be talking about the hand surface swab test. So let's begin with. So, mainly, we're going to analyze the following contents. Firstly, a brief introduction or sort of our aims that we have, then the steps that we followed in the process, followed by a discussion. Here in this part, we're going to take our methodology and we're going to see what things we had as our results. Then we have the final questions, which are no more than some ideas and some clues that we have to figure out about the practice that we've done. And then we have the results and discussions of the results. This final part is just the analysis of what we've done in the discussion. Like, for example, how was the number of bacteria found on the hands, for instance. And finally, a conclusion about everything. So let's begin with. First, we have the introduction. Well, it's important first to mention that as shown during 2016, he found that in urban transportation, on bus stations, train stations, these places that is usually where are many people just going, coming and going, coming and going. These places are so polluted by microorganisms. Specifically, that's because of hand-touched surfaces. As people are coming and going, as I said, they left microorganisms in those places. And besides, you can see that here 2018 also mentions that one of the largest human-associated microbial habitat is in the skin surface. What does it mean? It means that in our skin, in hands, or I don't know, or head, or part of our head, we carry a lot of microbial, which then can be propagated by ourselves. So, also, and the last part, which was so interesting to notice, was that, Fear did research, and in that research, he had to, he swapped two surfaces, he two hand surfaces, and then he noticed that they were like unique species, like in one place and the other they were the same microorganisms. So like so appealing to see this result. Well, so what are we going to do in this laboratory? What did we do? Basically, what we did was to do a surface swab test, hand surface swab test, which was done in order to identify the number of microorganisms present in the hands of the teacher, in also people who just have left the bathroom, and also in the containers, the food containers in the university. But specifically, our project or research was made out in the hands of our teacher, or teacher's hands. Yeah. So what we did just our goal here was to use the hand surface swab test in order to identify the microorganisms of our teacher's hand. Then we put those results, we put those swabs inside a tube test, and then we put them in a petri dish. Then we incubated the petri dish, and then we counted the number of bacteria in order to analyze the number of microorganisms present. Well, so let's continue. So what are going to be the materials that we needed? It was sterile swabs, which are no more than these things, the swabs that you use, a culture medium, and our plate count. So that's going to be like the medium that we're going to culture, we're going to put the bacteria to grow. Then we have the petri dishes. The petri dishes, these circle dishes. Then we have the sterile saline solution of peptide water, which is the medium that we're going to use to wet the swabs. Then we have the disposable gloves, lab gloves, masks, gloves, shoes. You cannot go to the lab without this without the allowed equipment. Then we have the incubator, which is going to be important for the cultivation of the microorganisms once they are in the petri dishes. Then a marker in order to put or to name each tube, each test tube. Then we have a burner, which is going to be to sterilize the place where we're going to work. And do not forget a piece of paper and a pen, because it's going to be important to draw the area that we're going to get a sample from. Well, so the methodology. It's very simple. I'm gonna do like imagining that I'm gonna do the test again. So imagine that this is the swab. Yeah. So first we have to, with a marker, we have to like to draw the petri dishes with the example. For example, since we're going to take the samples of two of my teacher's hands, so we put in the first petri dish and left hand plus the date. In our case, the date I think it was like the last day of April. I do not remember. It was a Monday. I don't remember exactly. But the thing is that we have to name both petri dishes. Then the next step is to wet the swab with the sterile saline solution. It means that with the solution that we have to put the swab into it so that this is gonna help us to, when we spread out the swab on upon our teacher's hand, what's gonna happen is that the microbial is gonna be they are going to be able we're going to be able to trap them actually. Yeah. So then we have the next step. This is no more than after we spread out the swab, then we have to put in a test tube, right? We put in a test tube, and important is to instantly after this process we have to ask our teacher to put his hand on the table so that we can draw it. It's gonna be important to calculate the area later. I will explain that later. Then the next step is going to be, as it says D, put the swabs of different test tubes with the solution. Well, as I said, after we spread out the swabs, what we did was to put the swabs into a test tube. Yeah, pure each. It means that we're going to have two swabs and we're going to put them into separate test tubes. E, we have to culture 1 milliliter of the solution of each test tube and insert them in a petri dish per each. It means that in the in the test tube that we had to take 1 milliliter of it, and then we had to put it into the petri dish. Yeah. So then, as we had put it in the petri dish, yeah, here I want to say my mention was to spread in the medium, to spread upon the medium. Yeah, next later we have to incubate the petri dish at 35 centimeters per hour. So it means that we have to to maintain that temperature. The petri dish is going to put in the incubator and we maintain the temperature. Yeah, then we analyze the results, which is the part that my partner is gonna explain. These are the final results. So Navi, is your turn.
Well, now I'm going to talk about the final results about the isolation. In this case, I wanted to explain the result, but I wanted to explain before the final results of the hand area. You can say in this slide the hand of the analyzed in the isolation. Before knowing the final results of the isolation in the teacher's hand, a drawing of the hand was made on a sheet or notebook where the squares of the sheet were taken and counted to get in the area where we gave us that the area of the hands is 38 square centimeters with 90 period by each side. In this case, you can say that this hand, this area is 13.8 centimeters square. But in this part, was divided by two parts. For example, in the palm, exists 21 centimeters square, only in this part of this palm. But in another case, the fingers, each fingers has a different area. For example, 18, 16, or 13. In this case, we use, or we were used this representation about the isolation. In this case, of the isolation, in the right hand, the area of each finger was considered of total area. Likewise, the area was looked at with the right hand, the same in this case as the area of the palm. In this draw, only to draw one hand, but it's considered to the right hand and the left hand. Sometimes that should be mentioned is that single drawing of the hand was used but considered, but in one single do not vary in area, or perhaps. Now, mm, now I'm going to explain the final results. In this case, in the Petri boxes, we have a different results. For example, in the picture of the left, you can see the Petri box that is the hisopation of the palm. Or for example, in the Petri boxes of the hisopation of the fingers. In this case, I'm going to talk about the hisopation in the left, in the right hand. The hisopated on the right hand on the finger, the samples of the hisopation carried out of the right hand, especially on the fingers. In the Petri dish with red agar showed as small colonies. You can see in this picture as small colonies that is so impossible to identify what is the UFC. And it's so complicated to form to microorganisms or bacterium with the range of for a total CFU was not reached, which led to the possible theories, where at the time of the blasting, it shows the agar was very fragile, which could inhibit its growth, as well as how to could be that the hand was legally washed, which meant that its growing was not reader. For another hand, in the hisopation of the left hand in the palm of the hand, was similar to the hisopation in the right hand. In this case, it's similar to the right hand, that is, but the right and left hands were not very contaminated or dirty, which prevent the growth of bacteria. But how um, how I said that it could be two possible theories, that is maybe the person of to help us it was washed hands, or maybe the agar is damaged or fragile in the preparation of the laboratories. Yeah. Our conclusion, we conclude that both hands in different parts of the same showed a very low growth of bacteria, which led to the conclusion that his hands were washed or disinfected after the hisopation. We don't know if this, we didn't know about this is real, about the disinfectation or wash it. That is so complicated to see the growing of the bacteria and preventing the growth. In addition, the hisopation was carried out inside the laboratory and not anywhere outside, or in a place where food is processed. Also, due to fragility of the agar previously reported by laboratorians. In this case, both spectrum was complicated in the solidification of the agar. The laboratory said that agar is fragile, for example, for the turn off the light, and complicate this possibility of to increase the batteries. And finally, to respond some questions about this hisopation in the laboratory. The first question is that what is the main objective of surveys swimming in the food processing plant's food? Well, the main objective of the surface waves in food processing plants is to detect and control the presence of pathogenic microorganisms and contaminants on the superfaces of facilities, equipment, and utensils used in food processing and handling. This is a main objective to implement this hisopation in areas or places that processing food. The second question is that describe the correct procedure for collecting and watch sample, because it is, and is it important to follow this method? In this case, a correct hisopation warrants the precision of the sample but will never give results. Two tests simple and must be prepared under aseptic condition and ensure that it is not accidentally contaminated with any other microorganisms. A selection of isopates where they can be breathed in unstained in a medium where we can be transported without danger or contamination to the laboratory. After selection, the sample is collected using sterile gloves. The moist swab must be removed on the superface to be sampled and to contact with other superfaces due to cross-contamination must be avoided. Finally, the transfer of the sample of the culture medium of growth from the laboratory, labeling and storage with relevant information. For example, the location and the time. Always, this is necessary to implement in the ground from the laboratory, and especially on the Petri boxes. And this method is so important because that warrants the precise and representatives of the sample collected on comparison of results and growing of the microorganisms and pathogenics. Something fundamental in the occurrence detection of pathogens and contaminants in food production environments. This is so important because detect if growing in products and avoid to these microorganisms to growing and damage in the people because it's so complicated to contain a air. And this method is very effective, and especially in processing food.
In this case, I will mention the question 3, 4, and 5. Firstly, we have name three types of surfaces that will be important to sample in a manufacturing plant for processing and explain why. Well, we have three important places that we can we can analyze. The first one is gonna be the equipment, the equipment and or employees. For example, if we can analyze their hands and also their clothes, because it's gonna be in contact all the time inside the food processing laboratory. Then we have the machines. They are also crucial for this research to be analyzed. Because the machines are the places where you are going to prepare that specific product. And if you find out that there are some that they are places which are polluted, there are microorganisms which are pathogens, it could be dangerous. And finally, the surfaces that are in touch with the food. For instance, if you have bottles or I don't know, pieces of cartons, and those pieces of cartons, I don't know, they are important for the food processing part, you have to analyze them too. Because imagine that you prepare a innocuous product, I don't know, a jelly. We call it the delicious jelly ever. And then you put this jelly into a bottle, a glass bottle which is polluted, and you didn't notice that because you thought, well, but I don't know, they were there and they were very well the last time that we analyzed them, but you didn't notice that there were there was a pathogen and all of those issues that sometimes happen. So therefore, later, there are gonna be problems, and that's not gonna be innocuous. The fourth question is the culture medium that is used in the practice, and which type of microorganisms can grow there. So for this example, we use just a general microorganism medium to culture. In this case, it was, as you can see here, a guard play count. What does it mean? It means that, well, since it's a general culture, there can grow bacteria such as aerobic and facultative anaerobic bacteria, as well as molds and yeast, as that. And if you are wondering why can yeast, molds, and bacteria can grow at the same time there, for that explanation we have the last question, which is explain the sample incubation process. Why are petri dishes placed into the incubator? It's for this problem. The thing is that if we don't put in the incubator, then what is going to happen is that the bacteria, the organisms that are in the petri dishes are not going to grow well because we need some rising temperature, like to have like a between 30 to 40 degrees Celsius, so that they can grow faster. So for those reasons, and as I have to mention my Mr. Matthew classes, we've learned that bacteria and other microorganisms can grow at a certain temperature. And therefore we incubate them so that they can grow faster. We can make the calculations, we can count the numbers later. So therefore, as you could have noticed in the laboratory practice, the slides, well, not the slides, like the guide of the laboratory practice, we incubated these samples in a 35 degrees, into 35 degrees, at 35 degrees. Why? Because at that temperature, the organisms that we mentioned beforehand can grow faster. That's the only reason. And well, so that's my explanation for now. And these are the references, the papers that if you have some time, you can read them. They're so interesting. And well, thank you so much. Hope you appreciate this video.